Thank you very much. All right, I think we will go ahead and take a short break and then resume at 1:30 with our presentations on the state of the science. So we'll go ahead and break for a few minutes.
Norbert, it's Patrick. I just thought I'd say hi and nice job thus far.
Thank you, Patrick. Thanks for joining. Look forward to your talk tomorrow.
I'm very much looking forward to it. It's been terrific thus far.
Great. All right. Okay, I think we'll go ahead and get started with session 2, which we've titled State of the Science. And we're primarily going to focus on 2 areas in this session, mechanisms of immune modulation, and then also we'll discuss some of these ionotropic receptors which are believed to be involved in pain and are being modulated potentially by cannabinoids and terpenes. And so with that, let me begin by introducing our first speaker, Dr. Barbara Kaplan. Dr. Kaplan is an associate professor in the Center for Environmental Health Sciences at Mississippi State University, College of Veterinary Medicine. Dr. Kaplan has been on the forefront of defining some of the immune modulating effects associated with cannabidiol, and has really been one of the leaders in this area. And also she has recently published a review on the immunological effects of CBD. So with that, I'm going to turn this over to Dr. Kaplan. Title of her presentation is Inhibition of Peripheral Immune Function and Neuroinflammation by CBD in EAE, which is an abbreviation for experimental autoimmune encephalomyelitis, which is a mouthful, but it's a model of multiple sclerosis. So with that, Dr. Kaplan, we will turn it over to you.
Okay, thank you. And thank you, Norb and the Center for Research on Ingredient Safety for the invite. I appreciate the opportunity to talk about our data looking at peripheral immune function and neuroinflammation in the autoimmune disease model that I'm going to talk about, and looking at what effects CBD will have on that. I'm not able to forward. Let's see. Okay, so I have no conflicts of interest to disclose. First, I'm just going to start about how we got interested in CBD in the immune system. And then, as Norb said, I'll be talking about our data in the multiple sclerosis-like autoimmune disease model. And then just some new effects at the end, where we have done some RNA-seq reanalysis data in order to help identify some targets. And we'll talk about what the significance of the findings are, and then acknowledge some of the people who did the work. So initially, my interest in CBD started when I was still working with Norb up at the Spartan MSU. It was soon after the time that the cannabinoid receptors CB1 and CB2 were cloned, and as Jin Peng showed, CB2 is highly expressed in cells of the immune system. So there was an assumption at the time that the cannabinoid effects must be mediated through CB2. And we kept getting all these pesky results that suggested that maybe some of the cannabinoid effects were not mediated through either one of those receptors. And so we wanted to use a variety of pharmacologic tools in order to help us understand this. So in addition to some knockout mice and antagonists, we started using CBD because, again, as we all know, it doesn't exhibit very high affinity for either CB1 or CB2. So we thought if it showed effects in the immune system, that would suggest that some of the cannabinoid effects are not mediated through either receptor. And in fact, what we found is that CBD looked very similar to THC, which does exhibit affinity for both CB1 and CB2. So there must be some non-CB1, CB2 receptor involvement. I'm just going to show you a few of those data that we established early on. This is just looking at one endpoint, which is interferon gamma, and this is a protein that's produced by a number of immune cells. And normally in mouse splenocytes, these cells will not produce a lot of interferon gamma in a naive or untreated state, but we can induce that interferon gamma with a number of chemicals or antibodies. So in this, on the left-hand side, I'm showing 4-ballester and calcium ionophore. This is a fairly inexpensive and not cell-specific way to stimulate those cells to start producing cytokine versus antibodies against CD3 and CD28 proteins that are part of the T cell receptor complex. So that allows that stimulation to be T cell receptor specific. But what we see in both of these cases is that if we stimulate interferon gamma in these mouse splenocytes, that CBD inhibits that interferon gamma production in a concentration dependent manner. We then went on to characterize the mechanism by which this was happening. And so in the top panel, we were still using the mouse splenocytes. And again, we stimulated those cells with the PMA and animycin, but now we were measuring interferon gamma mRNA production. And similar to what we saw on the protein in the ELISAs that I just showed, we saw that we could stimulate interferon gamma production quite robustly with the PMA and animycin, and that could be inhibited in a concentration dependent manner with CBD. So that led us to think that the mechanism by which CBD was inhibiting interferon gamma and other cytokines is transcriptional. And so we looked then at JERK T cells in the bottom two panels, and we used these cells in order to transfect them with a luciferase construct that contained either activator protein 1 or nuclear factor of activated T cell DNA recognition sites. And so that just allows the cells to have those sites activated so that you can tell whether or not those are targeted by whatever it is you're interested in. So again, what we saw in these studies is that under naive conditions, there's no activation of that construct. So the NFAT construct and the AP1 construct were relatively not activated, but that activation could be induced with PMA and animycin. And again, especially for NFAT, that NFAT-driven luciferase activity was inhibited pretty robustly by cannabidiol in a concentration-dependent manner. And it's interesting to note here because NFAT is a very well-characterized target of another immune suppressive drug called cyclosporin A, and we use that as a control. Of course, these were at concentrations that were about 100 times lower than CBD, so certainly cyclosporin A has more potency than CBD, but they are targeting a similar target in the cell. So these results from a few years ago showed us that CBD suppressed interferon gamma cytokine production in vitro, and we have a lot of evidence to suggest that other cytokines are also inhibited. And so the mechanism for suppression involves suppression of transcriptional activity, which leads to decreased gene expression. So that led us to the hypothesis that we started looking at when I moved down to the Bulldog MSU, is that CBD suppresses immune function, which will contribute to attenuation of autoimmune disease and neuroinflammation. As Norb said, we've been studying the model called experimental autoimmune encephalomyelitis, much easier said as EAE. This is an animal model that's used to study multiple sclerosis. And multiple sclerosis is an autoimmune neurodegenerative disease. So the advantage of this model is that it allows us to study immunity, whether that be peripheral or central, but also some of that neuroinflammation in a single model. So while the etiology of MS and EAE isn't totally understood, part of it is known to occur by the myelin that surrounds neurons being destroyed by immune cells that are recognizing proteins in that myelin. So if we look at a close-up of what comprises myelin, this is a depiction of a neuron that's being surrounded by a myelin sheath. The myelin is produced by an oligodendrocyte cell. And if we look here, here's the blue neuron over here, and this is the outside of the myelin, if you will. There's a number of proteins that are involved in creating this myelin sheath. And so what we can do in this model is we can use one of these proteins, and we use this one, MOG, which is myelin oligodendrocyte glycoprotein. And we use just 20 amino acids of that protein, and we emulsify that in a mixture with complete Freund's adjuvant, and that adjuvant acts as an immune stimulant. so that we inject that emulsified MOG plus CFA in the back of a mouse. That tricks the mouse cells into thinking that MOG is foreign. And so those immune cells will go and attack anywhere where there's MOG, which happens to be on the outside of the myelin. So what this does is that in the mouse, it induces a clinical disease that's similar to multiple sclerosis. It induces an ascending paralysis that starts with loss of tail tone and then proceeds up to single or dual hind limb paralysis. The disease can actually progress all the way to death, but we don't let the disease get that far. So we stop the disease no later than when the mice have developed dual hind limb paralysis. So we use this model in order to understand whether or not cannabidiol would be have any efficacy in the model. So on day 0, when we injected the MOG, that was our disease initiation day. After we create the emulsion of MOG and CFA, we inject that in 4 sites subcutaneously, 2 at the hips and 2 at the shoulders. And in this particular experiment, we had an interest in understanding whether or not CBD would affect the disease at 2 different levels or magnitudes, if you will. So we injected MOG at 100 micrograms, and we call that EAE. And then we also injected with 5 times lower the amount of that MOG peptide, and we called that mild EAE. So we weren't even sure at this point whether or not we would get difference in disease, and so that was another objective of this study. We then treated over the next 5 days with CBD, and we delivered that by oral gavage in a corn oil vehicle. And we give this to the mice at 75 mg per kg per day over that 5-day period. And then we necropsied the mice at 3 different time points. One was at day 3, relatively early in disease and in the middle of CBD dosing. The second one was at day 10, and then the third one was at day 18, which we consider to be our end stage disease. This is a point at which we have a high prevalence of mice getting disease in response to MOG, but before they progress to a moribund state, and some of them have the dual hind limb paralysis. So in looking at the data and whether or not CBD possessed any efficacy in this model, we showed that it could suppress the clinical scores in EAE. So as we see here, if we look at the saline CO control mice, they don't develop disease at all. The mild EAE did allow us to see a little bit of disease, but there really wasn't much there, and CBD really didn't do anything to that mild form of disease. However, the EAE at 100 micrograms did induce disease to an average of a clinical score of about 2. And in our hands, we define this as the ability of a mouse that can be flipped over on its back. Normally, mice will not let you put them on their back. But if their hind limbs are weakened to the point where you can flip them by their tail and they can flip over, that's a clinical score of 2. And so what we saw is that by day 18, there was a significant suppression of the clinical scores in EAE by CBD. And just remember that we had stopped the CBD back on day 6. So this is 12 days after the last dose of CBD. And I should also say that given some of the talks that were already shared this morning, this CBD is purified CBD that we obtained from the National Institute on Drug Abuse. So it's that 99% pure CBD. So it's just the single compound. In looking at the mechanism by which this happened, it's been well reported by us and others that CBD could induce some of these regulatory cells. And the function of the regulatory cells is, as the name implies, really to go and control other immune responses. We've shown this in vitro and others have shown it in vivo. We didn't really see any strong evidence to suggest that this was happening, at least in the peripheral immune system. So this is looking at either lymph nodes or splenocytes for a T regulatory population, or another regulatory cell called a monocyte-derived suppressor cell. And there are 2 different versions of this. So looking at, and these are both in splenocytes. So looking at this overall, we didn't really see much either with disease or with the ability of CBD to induce those regulatory cells. However, what we did see is that CBD could suppress some of the effector T cell function, and that is the ability of T cells to produce cytokine in response to the disease. So let me just say that over on the right-hand side here, when we're looking at IL-17, which is another pro-inflammatory cytokine that is produced either by a CD4 helper T cell or a CD8 cytotoxic T cell. Again, there really wasn't that much to be seen, either induced by the disease nor affected by CBD. On the other hand, we really noticed, and especially at day 10, which is about the middle of the disease, is that the disease state really was able to induce interferon gamma production, both from CD4 helper cells and from CD8 cytotoxic T cells, and that could be inhibited, and significantly so in the CD8 population, by CBD. Again, we noted that the mild state of the disease didn't induce as much interferon gamma, but again, CBD really didn't do much to that very mild form of the disease. And this significance of these results were that the CD8 cells in the periphery seemed to be more sensitive to inhibition than the CD4 cells. And also, again, this in the splenocytes peaked at day 10. We really didn't detect much happening at day 3 nor at day 18. So if we look then at what we just saw with the clinical scores, which really, we're evaluating later on, starting at maybe day 15 and all the way out to day 18, if we really focus on the pattern of what's happening at day 18, it really mimics exactly what we saw at day 10 in the spleen. So we have the disease inducing, we're getting induction of disease with EAE. We're also getting interferon gamma production and inhibition of disease, and we get inhibition of interferon gamma production. So one of our questions right now is what role, if any, is there for those peripheral T cells in the disease, since the disease really is a neurodegenerative disease and is likely manifested more at the target cell or at the target sites of the brain and spinal cord. But do these cells play a role? And that's certainly a question that we still have to answer. So moving on to the neuroinflammation endpoints, we first looked at some immunohistochemistry in the brain, and this is focusing in on the cerebellum. And at the top here, this is hematoxylin and eosin staining. And then the second line is CD3. This is the broad CD3 stain that includes all forms of CD3, all forms of T cells. The bottom is a higher magnification of that. So when we look at our saline and CO mice, we don't see a lot of T cells in the brain, and normally there shouldn't be a lot of T cells in the brain. But that can be really upregulated in the presence of disease, as indicated by these brown punctate spots. These are all CD3 stained cells, and that was really reduced by CBD. We saw similar effects in the spinal cord. Again, let me orient this. We have a lot of pictures here. The top is hematoxylin and eosin staining in the spinal cord again, and the second panel is CD3, and then these are the magnified views. So similar to what we just saw in the cerebellum, in the absence of disease, there's relatively low T cells in the spinal cord. That can be really increased with the EAE disease, and these are all at day 18, and then reduced by CBD. If we look at the CD4 positive helper T cells, which is one of the subsets of the CD3 cells, what we see is, again, low CD4 T cells in the spinal cord that can be induced with disease and reduced by cannabidiol. The CD8 cells were kind of an interesting story, and these were done with immunofluorescence. So there's very low CD8 staining in the spinal cord in the absence of disease. It's upregulated with disease, but really no appreciable effect by CBD on CD8 cells in the spinal cord. And this last panel was just an overlay of the CD4 and CD8 together, so we could look to see whether or not CBD was having an effect on perhaps cell-cell communication between CD4 and CD8 cells, but we didn't really appreciate anything there. So in looking at the quantification of the T cells in the spinal cord, first of all, focusing up on panel A here, this is just the total T cell count. And you can see it's trending in the right direction as far as inhibiting the neuroinflammation, but not quite significant. And then I'd just like to focus down here. We then quantified the CD4 and CD8 cells, not only counting the immunohistochemistry, but then isolating the lymphocytes out of the spinal cords, staining those first with a broad stain for lymphocytes called CD45, and then from there, parsing those out to either CD4 or CD8. So it was really interesting to see that the CD4 cells were, again, modestly inhibited or trending toward inhibition with no statistical significance, regardless of how we quantified these cells. And the CD8 effect really wasn't evident by either quantification. So that was kind of neat to see that the quantification looked the same regardless of how we did it. But again, those effects were pretty modest. So one of the last things that we did was we looked at GFAP, and this is a stain that can detect astrocytes, and when it's increased, that's indicative of activation of astrocytes. And so in this, we looked at GFAP at the top, DAPI, which is a cellular stain, and then the combination of the two at the bottom. And again, these are spinal cords at day 18. We see this ring of DAPI staining in the spinal cord in the absence of disease. And that is certainly lit up quite nicely in the presence of disease and then reduced by CBD. And the difference between these and some of the other figures that I showed is that this is the whole spinal column. And so with the DAPI stain, you can really see the column around the cord. So that's the difference there. So to summarize our EAE findings, we were able to induce different degrees of disease severity so that we could study whether or not CBD would have any kind of differential effect in disease, and that depended on how much MOG peptide that we used. So we saw that CBD was able to suppress the EAE disease, but really had no effect on that mild form, whether it be enhancement or inhibition. We noticed that the attenuation of EAE clinical disease at day 18 even occurred when we were giving CBD via oral gavage for the first 5 days of the disease process and then stopped. And then the mechanisms, although we don't totally understand everything involved with these, but it involves suppression of interferon gamma production from the spleen at day 10, which preceded the attenuation of clinical scores and that modest inhibition of several neuroinflammation endpoints at day 18. So in looking towards the future, we're considering using an RNA-seq method in order to understand what the T cells in the spleen are doing at day 10 under the disease state. I have a new graduate student in my lab, Krista Ferdella, and she has expertise in bioinformatics and RNA-seq. And so in addition to doing those studies that are coming up, she also performed a reanalysis of some published transcriptomic data that was performed on a data set in which a human neuroblastoma cell line was treated with CBD over time. So what they did is they just took these cells and treated with 20 micromolar CBD or the ethanol vehicle. And then they looked at several time points, 3, 6, 12, and 24 hours. So there was really a lot of data here. And so she went through and figured out where effects by CBD were happening over the time points. She, of course, identified a number of genes that were going up or down with CBD. But we're really interested in neuroinflammation. And so she manually curated these data and identified a few targets. And I'm just showing a couple of them here. 2 possible immune genes of interest. And again, remember, these are neuroblastoma cell lines, and this is in the absence of any kind of immune stimulation. So what we saw is that ICAM3 was downregulated at an early time point, and that downregulation was maintained over the time course, and that NOX01 was also downregulated early on, and then that really dropped off by the later 24-hour time point. And these are data in which CBD had effects as compared to its time-matched vehicle. So what are these targets? ICAM3 is intercellular adhesion molecule 3. This is a molecule that is involved in cell-cell interactions and binds to integrins and allows cells to essentially talk to each other. And NOX01 is a protein that can positively regulate NADPH oxidases, such as NOX1 and NOX3, and that can induce oxidative stress. So both of these are, I think, potential targets for not only the EAE disease state, but if CBD is inhibiting either one of those in our model. And that is certainly one thing that we're interested in. The other thing that we're doing is we're mimicking what was done in those neuroblastoma cell lines, but using the mouse splenocytes in a similar approach so that we can see whether or not some of these same targets are affected in an immune cell. And we're doing that both in the absence and presence of an immune cell activation. So we're looking forward to getting those results. So overall, we saw that interferon gamma and certainly others that I didn't have time to talk about was suppressed by CBD in vitro. And that mechanism involves inhibition of transcription factors that are important to drive the mRNA of those cytokines. CBD was able to attenuate the EAE disease process, And this involved suppressed interferon gamma from the splenocytes at day 10, which mirrored the clinical scores at day 18. And then there was this modest suppression of neuroinflammation with decreased infiltration of CD4 cells, at least modestly, and then decreased GFAP staining of astrocytes. And finally, our newest data suggests that CBD inhibited some inflammatory markers in human neuroblastoma cells, which are giving us options for future studies. So why are these data important? Well, as we all know and why we're all here, CBD is being touted as a wonder drug. If you just look at a few websites, you can see that they're suggesting that CBD is good for cancer, for your brain, for well-being, for spinal cord injury. So certainly the use and the exposures to CBD have increased dramatically over the last 10 years. And I mean, this audience certainly knows that. So there's a need to really understand its benefits and perhaps more importantly, its risks, and then to really understand the mechanisms by which it acts. And I think we can all agree that we need to keep talking about the science of CBD and get the science out there instead of some of these websites that are rooted in lack of science, for lack of a better term. So toward that end, I have tried to do some science communication about CBD, and as Norb mentioned, our recent review article, which we purposely published via open access mechanism, so that anyone who has an interest in at least a lot of the CBD immune data through the beginning of 2020 can look at that paper. But I participated in some interviews and scientific blogs on just about the science of CBD, and the CBD is certainly a big deal. We put out right after Epidiolex was approved by the FDA. So we're trying to get some of the science of CBD out in front of people. So with that, I certainly have to acknowledge many members of my lab. Much of the EAE data was done by Dr. Jim Nichols, who was a DPM Ph.D. who graduated earlier this year. He's now in a neuroimmunology lab at MD Anderson. And then 2 graduate students, one who is wrapping up, and then Christopher Dello, whose data I showed. And I had a number of students and undergraduates who helped me, and funding from NIH and others. And with that, I will take any questions.
Okay. Well, thank you very much for that, that interesting presentation, Dr. Kaplan. Maybe I'll start the questions, and I see a couple are coming in, so we'll give people a chance. So one question I had for you is I know that Savadex, which is not just CBD, but
Right.
it is a combination of actually 50% CBD and THC. And I know it's been used in various parts of the world for quite some time by people with MS.
Yes.
And I know primarily people are using Savadex to treat neuropathic pain and kind of relieve some of the muscle spasms associated with MS. But I'm curious if you're aware of any human data showing that, at least in patients using Savadex, if that at all is somehow inhibiting or decreasing the progression of MS.
Yeah.
I have a feeling there's probably a large number of people that are using Savadex, and has anybody really taken a hard look at that?
So yeah, that's a great question. And there are a few studies where people have looked at that, but there's not a ton of data out there. And the data that are out there aren't really that strong for Savadex inhibiting immune function. And I think there's, a number of reasons why that could be, not only pharmacokinetics or whatever, but some of the efficacy in humans may depend on whether or not the person is a cannabis familiar or not, and whether or not tolerance to some of the cannabinoid effects develops in humans, I think is a bigger consideration than it is in animal models, unless we can induce it on purpose. And let's see, what was my other point? I can't remember. So also Savadex is, again, could be pharmacokinetic because it's taken orally. But yeah, so, the last one is that when we're giving the CBD in the EAE model, those cells really, especially early on, are naive. They're naive to that immune stimulant. I mean, we give it the day after. And a lot of the studies that are done are with people who have multiple sclerosis. And if they're trying to study those specific responses to some of the myelin proteins, that might already be established. And we know that CBD and some of the cannabinoids aren't quite as good once that immune response has been kicked off. So very similar to how cyclosporin works, it really needs to be on board early.
Yeah, yeah. And just as a follow-up before I get to these other questions here, is I saw that you did some immunohistochemical staining of the brain looking at T cells, and I'm just curious, by any chance, did you also look at monocytes? Because, you know, the CD14, CD16 positive monocytes have been involved in a lot of these neurodegenerative diseases. And I was curious if you took that opportunity to stain for
Yeah,
them.
you know, we did not. So once we started down the IHC road, we really switched to the spinal cord and,
Okay.
you know, did all those studies there. So we really haven't. That's in our hands unexplored. But yeah, it's definitely something that we could look at.
Okay. Well, let me get to, I've got
Now,
one.
can I ask a question? This is Mitzi.
Oh, sure. Go ahead, Mitzi.
Barbara, I enjoyed your presentation. I have a question. In fact, I have two questions. One of them is to look, you found the double positives, the CD4 positive, CD8 positive T cells in the spinal cord of these mice.
Mm-hmm.
So I was just curious to know as to how, what do you think about those? Because in the periphery, usually double positives, unless they escape the thymic differentiation. So can you explain as to what you think is happening?
Yeah, so I'm not sure. We really didn't focus on those that much because it was relatively such a small population and there was no appreciable effect by cannabidiol on that population at all. So that was just, I mean, really, we did it for completion's sake because we had all the stains, but I don't know what role they're playing, if any, or, they certainly don't look that prevalent because we had the disease state. So I'm not really sure. We didn't really look into that.
Okay, and my second question is, I noticed that you were not, we had published on MDSC induction following CBD exposure and EAE,
Yes.
and I noticed that you did not find that. What do you think may be responsible? Because I realized you just mentioned that the dosing, there was something different from what is normally done. So could you go over that?
Yeah, so actually your data has puzzled me as well. So I don't know if it's administration because you're giving it IP. And so certainly our dosing is going to be different. I'm giving a higher dose than you normally do, but we certainly get a huge first pass effect. Years ago, we looked at plasma levels of CBD after the oral dosing, and it's not high in the plasma after even a 75 mg per kg dose. So I don't know if it's dosing or that you're delivering it right to that area where you're in the peritoneum. So I'm not sure, but yeah, I was kind of surprised not to see some splenic MDSCs as well. If anything, the Tregs in the spleen looked a little bit promising, but there was nothing significant. So yeah, but the MDSCs didn't look like yours.
Great. Let's see, I have a question here that's come in online. So the question, Barb, is given CBD's mechanism is at least in part suppression of immune cytokines like interferon gamma, would this suggest, or is there any data that chronic CBD use would make someone more susceptible to infectious diseases?
Yeah. So that's an excellent question. I think we don't know the answer to that. I think that hasn't been studied effectively. Jinpeng alluded to it a little bit in some of the clinical trial data with Epidiolex, and that there might be some increased incidences of either pneumonia or viral infection. And of course, I don't know from that table where those were. I'm not sure that those data are that strong if you really compare all the studies that have been done as compared to placebo effect and whether or not that's true. So for sure, that's an excellent question and one we don't have an answer to yet.
Great. Okay, let's see. Doesn't look like we have any other questions. So again, thank you very much for your presentation, and
Thank you.
we will move on.